Let's look at eukaryotes. Tiny eukaryotes. In this lecture, we're going to look at protists and define ecological roles. The objectives for this lecture: list and give 2 characteristics each for the 4 supergroups of eukaryotes. You may want to be able to give examples of each one of those groups. Define mutualism, predation, parasitism, commensalism, competition, and pathogen. And give examples of protists that fill each of the ecological roles. So what are the ecological roles? Well, we're looking at interspecific interactions, the relationships between species in a community. And we're defining this on how these interactions affect the population sizes. So a fox will have a negative effect on the population of rabbits, but the rabbits will have a positive effect on the population of foxes. This minus-plus interaction is defined as predation. Deer and rabbits both compete for the same resource, which means if there are more deer, there are fewer rabbits. And if there are more rabbits, there are fewer deer. So this is a minus-minus interaction. We can lump these together in this lovely grid. You see the plus plus is mutualism if it's a beneficial interaction between both organisms. A minus plus is your predation, also herbivory because more rabbits means fewer dandelions, and parasitism is also a minus plus. That minus minus is interspecific competition, which we define as different than intraspecific competition. A 0 plus is commensalism. Think of lichens living on Douglas fir trees. This does not affect the Douglas fir trees in the least, but the presence of Douglas fir trees is very positive for lichens, giving them a habitat. Your 0 minus is amensalism. This is where the presence of one organism has a negative interaction on the other without affecting the first. So one example of this is a certain type of goatfish, which stirs up the sediment in the bottom of the ocean, thus making it harder for visual-based predators to prey on things and having a negative effect on their population. And 0,0 is neutralism. It's not an interaction. It's the lack of an interaction. We're going to look at those interactions as we look at protists and how they affect each other and how they affect other organisms. So what is a protist? Well, this is a paraphyletic group. It's any eukaryote that is not a plant, not an animal, not a fungus. Most protists are unicellular, some are colonial, like the one shown here, and some of them are multicellular. This is your definition from your book. So this is all eukaryotes except what we're going to look at in later lectures, the plants, animals, and fungi. And many ecological roles are taken by protists, and they are divided into 4 supergroups. I want you to look at this phylogeny, and you can see plants are in the Archaeplastida. But plants are different than protists, so we don't count them in that paraphyletic group. Fungi and animals are in the Uniconta, but fungi and animals are not protists, so they are not in that paraphyletic group. Then we have the excavata and the SAR. So how are we defining these? Well, the excavata, we're going to look at some slides with those, have a groove excavated on one side of their body. The SAR, that's a lot of different things. It's 3 separate groups for the price of 1, including many photosynthetic organisms and examples of secondary endosymbiosis in there. The Archaeplastidae are the red algae and green algae, and the Uniconta are amoebas and other things similar to those. First, let's look at the excavata. There are 3 examples given here. First off, the diplomonads. They have reduced mitochondria and use anaerobic respiration. Giardia is one example of this that is a parasite. Parabacillids also have reduced mitochondria, and they actually produce hydrogen gas. Trichomonas is another parasite that is in this group. The euglenozoans are made of predators, photosynthesizers, and parasites. They have things called kinetoplastids in them, which we'll look at a little more in lab. So some things that are in the excavata, you see a groove on the side, and they have often odd mitochondria. Many of these are parasites, but not all of them. Obviously, euglena is not a parasite. For the SAR, we're going to look at 3 separate groups in here. First off, the stromatophiles show an example of secondary endosymbiosis, which we can see in having 3 or 4 membranes around the chloroplast. Some examples of these are diatoms, and they have glass-like walls and this organic matrix made up that holds their body together. And they have, of course, chloroplasts in them. Diatoms do a lot of photosynthesis out in the open ocean. Closer to shore, we have the brown algae, which arose in the Jurassic. They're actually the youngest type of algae. They are multicellular and they're mostly marine, and they are photosynthetic organisms. The alveolates are the A in SAR, and they have these membrane-enclosed sacs, think alveoli, under their plasma membrane. There's possible endosymbiosis in this group, and some examples here are the dinoflagellates. They have these two flagella in grooves and kind of spin around. They're an oceanic algae. The ciliates, these have cilia. Think about paramecium here. And they're often predators on bacteria and other small organisms. And the apicomplexans have this complex little apical anatomy, and many of those are parasites like malaria. So these are three groups in the alveolates. The rhizarians have these complex shells. And you can see forams here with these shells that have pores in them. And cercozoans have these amoeboid and flagellated protists in their groups. So it's not as well defined by features as the other two groups in the SAR. Archaeplastidae, or essentially old plastid, are the things that gave rise to plants. So we think of plants perhaps as the new plastids because they have chloroplasts and evolved from the old plastids, these Archaeplastidae. They are photosynthetic. The red algae have this wonderful reddish pigment called phycoerythrin, which makes them red. Some examples are Bonomysonia hemifera and, of course, our sushi nori. Green algae are common photosynthetic organisms like sea lettuce that you can find in many ponds, lakes, and the ocean. The uniconda are the amoeba, fungi, and animals. They've got these lobe or tube-shaped pseudopods if they're amoebozoans. The ophisticons are animals, fungi, and some other protists. We're going to look at animals and fungi extensively later. These organisms are heterotrophic. They do not photosynthesize. Looking back at interspecific interactions, we can look at examples of how the different protists fulfill these roles. So we're looking at parasites pretty often with those excavata. And of course, when we start looking at SAR, we realize we don't see a spot of those where we put them, so we're just going to lump them in photosynthetic. It's not really an interspecific interaction so much as something they do. They are the things that herbivores eat. And we've seen some examples of the excavata in parasitism, but we also see those ciliates being predators and even herbivores if they're feeding on plants. In class, we're going to look at the different protists in terms of how they interact with other organisms. I would recommend you do this on your own. Remember, if it's a disease-causing parasite, it's called a pathogen.